most of the time it's fine, but depends on the situation whether how much accuracy difference across the subjects. If the difference or the reliability changes a lot across subjects, then we may face some issue. So that we can, because we're doing whole brain, it's not just one voxel. So I cannot say, depends on the region in the brain too. So it's difficult to say ahead of time what's the exact impact. So unless you have to try the two approaches, then say the difference. So that's basically a scenario. So in theory, of course, it doesn't hurt you. Even if there's not much accuracy heterogeneity across subjects, the approach still works fine, right? It's just whether you weight, the weighting would be treated equally if there's little heterogeneity across subjects. So theoretically speaking, this is always a good way to do it, to adopt. But the more practical issue is whether you want the computation cost and also the modeling complexity. So that's something you have to keep in mind. So now let's switch to the specific fMRI programs. Which scenario you would use which program? So that's basically I call it roadmap. So there are many factors that you take into consideration when you decide which program to use. So first, I want to mention how many programs we have for group analysis. There are quite a few. First of all, there's the t-test, which is the t-test. The program is called the 3D t-test plus. Why it's called the plus? Because there's an old program called the 3D t-test. That program is already obsolete, so we don't need to mention that. So 3D t-test plus, as the name indicates, it's a program that handles the simple t-tests. Use that program, you can basically do one sample t-test, two sample t-tests, paired t-tests. You can also use it to do basically general linear model. So if you have a quantitative variable, you can do regression. So you can do simple ANOVA. So you have two groups, for example. You can two groups plus covariance, like age, IQ. You can throw in. You can do general linear model, right? So that's even if it's a paired t-test plus a covariant, you can reduce the paired t-test into one sample t-test. Then you can use 3D t-test plus to handle that. Two conditions. You just get the difference between two conditions plus the covariance. So that program can handle actually more than just as the name indicates, the t-test. You can also use sometimes to do a general linear model, even paired t-test plus the covariant. So that's the one scenario you have t-test. Another scenario you may think about is 3 t-test plus has the embedded functionality of doing multiple testing correction embedded, which I will talk about this afternoon. So that's other programs like ANOVA, 3D MVM, 3D linear mixed LME linear mixed film approach. The multiple comparison correction step has to be a separate step. Unlike 3TS++, the program itself can directly perform the correction step. So that's another advantage of using 3TS++. So even if you have a ANOVA scenario, because of that advantage, you may break your ANOVA structure into multiple t-tests. Then you use 3TS++. So that's something you may take into consideration. So that's a t-test to do a t-test or to a general linear model. If you have ANOVA, then basically you can use 3D MVM. That's a multivariate modeling approach to handle all sorts of ANOVA structure, right? So in fMRI, the ANOVA, we don't have the problems of dealing with the incorrect F statistic formulations. And the t-tests are also, post-hoc t-tests are also handled properly. So whatever your ANOVA structure or ANCOVA, as long as it's largely within subject ANCOVA, you can use 3D MVM. So the last program is a 3D LME, linear mixed effects model. That is usually you use that to handle some peculiar situations like Bayesian data, or you have within subject covariant. Right? Those are the two popular scenarios you may consider using 3D LME. Another situation is that if you want to calculate the ICC intra-class correlation, you can use 3D LME. to compute intra-class correlation. We also have 3 ANOVA programs that probably, for simplification, I will just ignore that because nowadays, I mean, I just recommend you use 3D MVAM. Unless you are familiar with, you are already a veteran FNE user, you know how to use those programs. I will briefly to mention them as well later on. So first started with 3 t-test plus, as I briefly mentioned before. It's, I forgot, got also mentioned 3D MIMA. That's pretty much the mirror image of 3 t-test plus, except the major difference is 3D MIMA, you need to take both beta and the t statistic as input, right? So that's the major difference. So we use that to handle t-tests, to handle a univariate general linear model. But you can also handle simple ANOVA structure like 2x2 or 2x2x2 even, as long as you know how to break the ANOVA structure into multiple t-tests. That's especially the interactions. It's a little bit tedious, but as long as you know you get the hang of it, then you can still use 3 t-test plus to do it multiple times instead of just once. So that's the 3 t-test plus. Then multiple scenarios are ANOVA. I see here there's a 3D ANOVA, 3D ANOVA 2. I mean, we can just ignore, just simply use 3D MVM because that's easier to understand and handle it. So there are multiple different ANOVA structure. I mean, it can be purely between subject ANOVA or purely within subject, or can be the mixture of 2 types. All right, so let's look at one example. Here, this is the semi-experiment design Rick already mentioned yesterday or even Monday. That we have 2 conditions, right? So that's auditory condition versus visual. So reliable visual versus reliable auditory. So the first script, it's called we use 3 t-test plus to handle one condition, to do one sample t-test for the reliable visual condition, right? So, and it's this standard FNE format to write a script. So first we have the program name, then we use the prefix to specify the output file name. Then if you want to mask out the outside the brain, you can provide a mask. Then you just list all the input files. In this case, remember we just focus on one condition, so 10 subjects. So one group of subjects plus, I mean, each subject has one beta, which is the reliable visual. So you may notice that in FNE, the file from individual subject output, analysis output, the file contains its 4-dimensional data set. So the file contains 4 dimensions. The 4th dimension is the in this case we call the term is called a sub-break. Each one is a break. It's a 3-dimensional. So that can be each 3D data can be beta value, can be T statistic, can be F statistic, can be correlation value. So because here we want to only take the beta associated with one particular condition, which is the reliable visual. So we need to specify, we call it sub-break selector, use the square brackets to specify which beta we want to use as input for 3 t-test plus. Now you may notice here within the square brackets, I use the labels instead of a number. Either one would work, but I strongly recommend you use the label. Why? Because the label is self-revealing. Immediately you say the label, that's because as a user, you designed it. You put that label in. You know what it is. Unlike the sub-break number, the number is just a number. If number 5, that doesn't tell you much, right? Not as informative as the label. That's one reason. Another reason, it's more. I mean, it's less. You will not be able to make a mistake if it's a number. I mean, some subjects, the output file from individual subjects may be different across different subjects, right? So that may may cause some trouble. So that's another reason I strongly recommend to use the label instead of subject numbers, subject numbers. So it's always the label attached with a pound sign, then the 0 underscore coefficient. The coefficient, that means it's beta value. So that's pretty straightforward. So how about we use the mixed effects modeling approach? Take both betas and the t-statistic input. That's the second half on this slide is 3D MIMA. Pretty much this similar structure, but instead of one set of input file, we have two sets. Remember, so now the second set is the t-statistic. As you can see here, there's this appendix part, the underscore t-stat. So that's so the label also in this case is more informative than the subject numbers. One more thing I want to mention here is the last line in the 3D MIMA script is that putting missing data 0, that is important for those voxels along the edge of the brain. When you have a mask, that mask maybe they're slightly different across different subjects. The mask maybe for this subject, for this particular voxel at the border, the next subject maybe does not have the data, right? So if you have 10 subjects, maybe only 7 of the subject has a beta available. 3 of them maybe have a number of zeros. So when you do group analysis, for that particular voxel along the border of the mask, you only have 7 subjects available. If you don't do something special, then just take the other 3 subjects with value of 0. Then you can do. You are going to. I mean, the program will work. Of course, will not say complain. But the problem is the analysis is not accurate because you have missing data basically. So how to handle that is basically you tell the program, whenever you say there's a beta value of 0, consider you have missing data. Then you can do group analysis. Only focus on those subjects with real values. The zeros values, for the subjects with 0 values, we're going to treat it as missing data. So that would provide a more accurate modeling for the group analysis. Any questions about this? So that's 3D t-test++. So similarly, actually, for 3D MIMA. For 3D t-test++, there's a similar concept. It's called the option is called z-skip. It's exactly the same concept, just a different option name. So that's one sample t-test. Similarly, for 2 sample t-tests, which basically you have 2 groups. Paired t-test, that's you have one group of subjects, but 2 conditions. So here is an example using 3D t-test++ to handle paired t-test. That's still the exactly same data set. We just to perform the difference between the 2 conditions. It's a reliable visual versus reliable auditory. Right? So that's why we have 2 sets of input. So we use set A, set B, those 2 options to specify the 2 conditions. Plus, we need to add the option paired. If we don't put that option, it will treat those 2 sets as 2 groups. So the default, if we don't put the paired option, you would the program will treat it would do a 2 sample t-test instead of a paired t-test. So you have to be very careful. So if it's a paired t-test, don't forget to put that option. We can do something similar with 3D MIMA, except that we have to put the contrast as input because the program cannot handle the 2 conditions as separate input files. So that's why we provide, notice the label here. We provide the contrast, the beta for the contrast as input, as well as the t-statistic for the contrast. Right? So that's, other than that, it's pretty much similar for the one sample scenario. So that's a t-test scenario. Then ANOVA, I mean, there are several scenarios about ANOVA. So first we have the one way between subject ANOVA. That means just have one factor. It's a between subject factor. That's 3D between subject ANOVA. Then we'll have multiple way between subject. That means we have more than one factor but that all the factors still are purely between subject factors. So let's switch to the another end. It's one way within subject factor, ANOVA. So we have one factor. That factor is within subject. That means like two or more conditions. So that's ANOVA. Or so here is an example we can use. Even though it's a paired t-test, we can treat it as an ANOVA because paired t-test is ANOVA. It just have one factor. That factor has two levels, right? So it's just a special ANOVA. So we can use the same paired t-test scenario to use the ANOVA structure. So in this case, it's an older program called 3D ANOVA 2. But you can use 3D MVM 2 as well. So it's just different ways to conceptualize the data structure and use a corresponding program. So we can keep going two-way within subject ANOVA, then two-way mixed. That means you have one between subject factor plus one within subject factor. So that's mixed. So now let's come back to the demo example I mentioned before. In that case, so in this case, we have, remember this is a 2 by 3 plus the covariant example I showed at the beginning of the demo example. So then in this case, remember we have first factor is a between subject factor. We have two groups. We have patient versus control. The second factor is three emotion conditions: positive, negative, and neutral. So two factors plus we have a quantitative variable is age. So 2 by 3 plus age. So the traditional approach in neuroimaging, when they use univariate general linear model, they would not be able to handle this because of we have the age variable in the model. The traditional univariate general linear model would not be able to handle it. But if here we are using multivariate model approach, so then it's pretty straightforward. So the program is script is a little bit different from the typical other programs like 3T++, 3D MIMA, or 3D other ANOVA programs. So here we need to put the data table, I call it data table, at the bottom of the script. So the table is basically lays out the data structure. So the first column is the subject labels. You put, that's subject, the ID, for example. The last column is the input files. In this case, the LIFT format, right? So NII, that's a LIFT format. So remember, the first column is the subject IDs. The last column is the input files. In between, you just put all the explanatory variables. What is a factor, either factors or quantitative variables or covariates. So in this case, we have three columns, we have three variables, three explanatory variables. We have two factors, plus we have a quantitative variable. So the second column, that's a group. So basically either it's control or it's patient. So this column condition, that basically says each beta is either positive, negative, or neutral. So this column in the middle, that's age. You put the numbers, the age values for each subject. So that's the data table. So that describes the data structure. So the first line, that's 3D ANOVA program, the program name. Prefix, we specify the input file names. Jobs, we use that option to specify the number of CPUs you want to use, because this program usually takes much longer time than 3T++. So we need to take advantage of a parallelization. So use the multiple CPUs. So then the next two lines, it's a little bit esoteric here. So look at let's first describe the second line, BS vars. That BS means between subjects, variables. So in this case, we have two between subject variables. First is group, right? That's basically it's patient versus control. Then we have age. Age is, also, even though it's a quantitative variable, it's a covariant, but it's also between subject variable. Why? Because each subject only you just have one number, so that's between subject variable. So also you notice there I put a star there. That's R notation. That means we want both main effects plus the interaction. So A star B, the variables A and B, A star B means A plus B plus A colon B. So that means we want A, main effect of A, main effect of B plus the interaction between the two. A colon B, that means the interaction between the two variables. So the next option here, WS vars, we use that option to specify within subject variables. So here we only have one, so which is the condition. So we have positive, negative, neutral. So those three conditions. So that variable is a factor, so that's a within subject variable. The next one, Q vars, that's a quantitative variables. So in this case, we have age, that's a quantitative variable. So that's basically covariant. Then the next few lines, basically, if we stop there without the next few lines, basically we're going to get all the omnibus tests, the F tests, main effects plus interactions. That's it. But that's usually not good enough for us. We need to use the t-tests to partition out those omnibus tests. So that's why we need to use those few lines to specify the exact post hoc test we want. For example, the first line here, we use this general linear test, GLT, general linear test, GLT label. The first number is basically index. Then you put a label. The label basically is an indicator what the test is about. So then we use GLT code to code exactly what we are looking for. For example, let's look at the code first. So we want to control the group at the patient group. So the group, first you have the variable name, space, colon. After the colon is a space, we have the weight. I mean, usually the weight is 1, either 1 or minus 1. So we want to fix the group at the patient group, then also fix the condition at the positive condition. So that's why here is in both cases the weights are 1. The weight is 1. So that's very boring, right? So we want to basically say the combination of the group and the condition. So basically we want to say the positive condition of the patient group. So that's the first post hoc test. The second one, we're looking for the contrast between positive and negative for the control group, right? So for you, but the first time it's a little bit overwhelming. It's difficult to decode this, but unfortunately, but we have if you write the script, you probably have to take a little bit time to learn the specific. But we have GUI. It's called a 3D MVM validator. That GUI, the only thing you need to do is create this table underneath. So then the GUI will help you to not the right way the script like this, but it has the buttons that you can use to specify exact those options and the post hoc tests. So but for beginners, probably that would be a better way to start with. Once you know it, then probably it's easier to direct write the script. Probably much faster to use the GUI part. So that's basically the structure of the script. So it's because like the options like between subject, within subject, and quantitative variable. So those are something new. That's something you need to get used to it. So you need to specify the variables. The program will automatically, based on the information provided, to generate the model. So in this case, basically the model is the group by age by condition. So you are going to get all the possible interactions among those three variables. You're going to get a three-way interaction, two-way interactions, plus all the main effects. There are three main effects. So that's basically what how the 3D MVM works. Now let's switch to another scenario. Is that so far we only talk about the situation. Each condition you have one beta. That means we model the hemodynamic response with one curve. right? As I mentioned Monday, we make a strong assumption regardless what task, regardless what subject, regardless what brain regions, it's the same curve, regardless what curve you use. That curve is fixed. So if we want to do a little bit fancy, we don't throw away that assumption. We just use the data to estimate the hemodynamic response shape. So then you have with multiple betas. Suppose you use 10 tenths, right? 7 tenths, for example. You end up with 7 betas. So what can we do, right? So that's the challenging at the group level. We have multiple betas. That's one scenario. Another scenario in SPM, you have those adjusted, I call it adjusted shape approach, that you have 3 basis functions. So the typical SPM approach, you throw away the second beta and third, those 2 betas. You focus on the first beta, the canonical curve, canonical response curve, the first beta. So that's, you just do it the typical approach. So can we do it better? Or also how do we handle the multiple betas at the group level? The simplest approach is basically, I think, let's just ignore the complexity. Just treat those multiple betas as a factor. So that's the simplest approach. Just suppose you have 7 betas associated with those 7 10 functions. You just call that an extra factor. It's a within subject factor, of course. Whatever you want to call it time or call it basis functions, you could just put a label as an extra factor. So then you may ask, what kind of a gain would it give us, right, if we treat that? So here is an example. There's a better example I mentioned Monday, it's to demonstrate this modern approach. So the first row is just, we use multiple betas. In this case, we have 10 functions. So we have 2 conditions in this example, and plus we have the H as a covariate. So actually it's a 2 by 2. It's a child versus adult. So that's first, it's a between subject factor. Then I have 2 conditions, the congruent versus incongruent. So I have 2 factors, one within, one between, plus I have H as a covariant. In addition to that, each condition is either congruent or incongruent in beta values because we use the 10 basis functions. So we compare 2 approaches. First approach, we use we estimate the hemodynamic response with 10 betas, with 10 basis functions. The second approach, the second row, we use the SPM approach. We use 3 basis functions: canonical curve, time derivative, plus the dispersion curve. So those are those 2 approaches. So in the end, when we come to the group level, the approach I mentioned before is that we just treat those 10 betas as a factor. So in the end, whenever we say something about the effect, we basically look at the interaction of that multiple betas with the other whatever effect we're talking about. So in this case, we look at the interaction of those 2 factors, the interaction between group and the condition. So usually that's a 2-way interaction, right? 2 by 2. So that interaction, in the traditional approach, if just 1 beta, we would look at the 2 by 2 interaction. However, here, because we have 10 betas, so that originally that 2-way interaction becomes a 3-way interaction. So we look at the interaction between those 2 factors and the 10 betas. So that would be the 3-way interaction. That would be this part, this image. Versus the approach, the SPM approach would be on the right-hand corner, this one. So that's the SPM approach. You only take the first beta, ignore the time derivative beta and the dispersion beta. So you focus on the first beta, the beta 0. So that's how much you see versus we use the estimated hemodynamic response. So that's the big difference. You may see here this blue circle here That's you see it in the SPM approach, but not in this approach, missing. However, that missing part can be compensated by this. There's extra information available through, look at some main effect that can be found through a complementary approach. So this is more powerful in a sense. You're going to see more clusters, way much more than the traditional approach. You're going to detect more. Why is that? Well, not just because in the end you look at the clusters. You are also going to say something about the excited response curve. Unlike the traditional approach, you just have one beta. There's nothing you can talk about the curve, right? Here, because we estimate the response, you can immediately use to plot out the response function, the shape. So here, let's look at specifically what those curves are. So the first column are the two groups. The first group, focus on the first group. The two colors code for the two colors. So the first column is for the adult group. Second column is for the child group. So the two colors, the blue one is for the congruent condition. The red one is for the incongruent. So look at each one. We can see, for example, the first one, the magnitude, the peak is about the same between the two conditions, congruent versus incongruent. But the difference is about the recovery, the undershoot part. Of course, the traditional approach would not work. The SPM approach would not help either because in the end you throw away the adjustment. You focus on the peak. The peak will not help you to detect the subtle difference like the undershoot part, right? So even for this, the second scenario here, there's an undershoot for one condition at the beginning. This undershoot is right after the onset, before the peak. Undershoot, the traditional approach will not help you to detect either because that subtlety, you have to use this approach to see such nuance. So that's, there's many other scenarios like here you have both undershoot at the beginning and the end as well. So that's why this direct estimate the hemodynamic response shape would help with getting us detection power. How we handle the scenario where you estimate the hemodynamic response shape. So then there are other complications, complicated, but I can ignore that. Trend analysis. Covariant. So I want to say a little bit more about the covariant. The covariant, one issue is covariant is, there are several scenarios. One is you may have outliers, but it's always complicated how to handle outliers. But let's ignore that. One more thing. One specific thing I want to talk about the complication of modeling covariant is that so-called centering. Why the centering is important? This is theoretically speaking, the centering is not really such a big deal. Textbooks usually don't even discuss it. It's more a practical issue. The reason is it would have impact on the interpretation of the result. Why? Well, let's look at whenever we do a regression, covariant is in here. I specifically use it for the scenario where the variable is a quantitative variable. For example, in this case, we talk about the age. We want to use the age as a covariant. Suppose at the group level, we just have one group of subjects, just one condition. Why is centering a big deal? That's because when we fit the IQ, not the age, the IQ as a variable, we just simply fit it as a straight line. Whenever we have a straight line, we need two parameters to specify that line. One is the intercept, the other one is the slope, right? So that's how shallow or how steep the change between the brain response and the IQ is. So one is the intercept, one is the slope. If you're only interested in the slope is, I call it a marginal effect. That means when IQ increases by 1 unit, what's the amount of the brain response would be. So that's basically the marginal effect. But the intercept, what is the intercept? Intercept is when the IQ is 0, what's the brain response? Well, that sounds like very strange, right? Nobody would be interested in the brain response when the group average is IQ is 0. Nobody would be interested in that, right? So that is a problem. But the well interested is when the IQ is at some particular number, like group average or the population IQ, then we want to know the average brain response. So that's what the central issue is about. So how can we do that? How can we achieve that? That's the crucial question. That's the centering part. How do we do that? That's basically